Hello, this is Davina Teaches Biology, back with another video. Today, an introduction to nucleic acids topic. The nucleic acids are DNA, RNA, and the one that throws students off a little bit, ATP. All of these nucleic acids have the same monomer. Monomer means single repeating unit. The monomer, which will be repeated, is the nucleotide. A DNA nucleotide has this basic structure. We have a phosphate group, deoxyribose sugar, and a nitrogenous base, because it contains nitrogen, sometimes also called organic base. RNA nucleotides have a very similar structure. Phosphate group, ribose sugar this time, not deoxyribose, and nitrogenous base. ATP is known as a phosphorylated nucleotide because it has the basic nucleotide structure but additional phosphate groups attached, therefore making it phosphorylated. We have 3 phosphate groups, ribose sugar similar to RNA, and just 1 nitrogenous base. In DNA, our nitrogenous bases can be adenine, thymine, guanine, and cytosine. In RNA, the thymine is replaced with uracil, but adenine, guanine, and cytosine remain the same. In ATP, the only base is adenine. The others are not present. DNA is made of 2 polynucleotide strands. Polynucleotide meaning many nucleotides joined together to form a polymer. And if I were to zoom in to those polynucleotide strands, we will see 1 DNA nucleotide, another DNA nucleotide, joined to another. So this repeats to form the familiar polynucleotide strands seen in a DNA double helix. Double meaning there are 2 strands which are twisted together to form this helix structure. Each individual nucleotide is joined to the next nucleotide by bonds. These bonds are phosphodiester bonds, the deoxyribose sugar of 1 nucleotide forms the bond with the phosphate group of the next nucleotide. This reaction is a condensation reaction. If you remember from the beginning of your A-level course, you would have learned that during a condensation reaction, a bond is formed with the elimination of water. In order to catalyse this condensation reaction, the enzyme DNA polymerase is used. So I have drawn just one side of the double helix, one of the strands. You then have to imagine that there will be another polynucleotide strand, but it will run in the opposite direction. because the strands are actually anti-parallel, which means in one strand, the direction is this way. In the other strand, the direction is the opposite way. In my next video, I will talk in more detail about the structure of DNA and how it is replicated. RNA is a single polynucleotide strand. So there are not two, just one. However, each nucleotide is still held together by the same phosphodiester bonds. To catalyse the reaction, we need a different enzyme, RNA polymerase. ATP remains as a single molecule. It does not join to other nucleotides, so it is not a polynucleotide strand. This was the basic introduction to nucleic acids topic. Watch my next video so you can hear the detail of DNA structure and semi-conservative DNA replication. I have already made a video talking about RNA and protein synthesis, so make sure to watch that one. Please like my video, add a comment and subscribe to my channel so that I can add more videos to help other people with biology. Bye.